Hi, my name is Cameron, and my thesis investigates the effect of exosomes on cystic kidney cell proliferation. So just first, a little background. When I'm talking about cystic kidney cells, I'm actually talking about a very particular disease called autosomal dominant polycystic kidney disease, or ADPKD. It is the most widespread inherited kidney disorder in the world. I actually have it myself, in fact. It's really interesting because it is caused by a single mutation in one of two genes, either PKD1 or PKD2. And a mutation in one of these genes causes the formation and growth of renal cysts throughout one's lifetime, from the time that they're in the womb to the time that they die. And in 50% of all cases, there is eventual kidney failure, I think by the age of like 50 or 60. Currently, there are no cures for this and very few treatments. In particular, I'm looking at the effect of the intersection of exosomes in PKD. Exosomes are, like we just saw, extracellular vesicles, the little membrane-bound particles that have varying components inside that are shipped out of one cell and brought into another. They're very important for a lot of different things, like cell signaling, material transport, and there's also evidence that they're very important for the progression of cystic diseases, as cystic cells export exosomes with unique contents, proteins and RNAs that are brought into healthy cells and cause them to exhibit more cystic behaviors, more proliferation, different metabolic pathways, et cetera. There's also been some research that has shown that exosomes from kidney epithelial cells do actually contain these two proteins that are implicated in PKD. This one study from Ding et al. Showed that if you take exosomes from PKD1 null mouse kidney cell cultures and incubate them with normal mouse kidney cells, you see both a decrease in the expression of these two PKD genes, and then also an increase in cell proliferation, both of which are suggested that cystic behavior in these normal cells is increasing when you put these cystic exosomes on them. And they've also shown that if you take exosomes from the urine of the kidney disease patients, urinary exosomes are just kind of the best way to get these specific types of kidney exosomes. It actually has the same effect on the mouse cells. It also increases their cystic behavior like proliferation. There's still some unanswered questions, though. First of all, do exosomes from PKD patient urine also promote this proliferation in human kidney cells? Because prior research has mostly been on mouse cell cultures. Also, what about in kidney cells that already have this PKD1 mutation? So what about cells that already serve as a model for the patients with this disease? So if we're looking for like a therapeutic effect, we want to assess the effect of these exosomes on cells that mimic the actual patient's cells. This leads to my hypothesis, which is yes, in normal and PKD1 mutant human kidney cells, urinary exosomes from PKD patients will promote greater cell proliferation than exosomes from normal individuals. The experimental design basically has two different phases. First, we just want to isolate and characterize our urinary exosomes to make sure that they are quality and act the way that they're expected to. And then we want to actually do our experiment and assess the effect of the exosome treatments on cell proliferation. So for phase one, for isolation, we have a pretty simple process. It's just a sequential centrifugation process, which basically just means we take the urine, we centrifuge it one or two times to pellet out the debris and the larger proteins that we don't need. We get rid of those and collect the supernatant, and then we spin that at 150,000 times gravity, and that pellets all the exosomes into a little pellet that we can suspend and have our nice exosome isolation. Then we can characterize them. There's a few different ways of doing this. First of all, we can quantify them using nanoparticle tracking analysis that we just saw in the last presentation. And this is basically where we take a small sample of these cells, pass them under a really high-powered video microscope. It collects different videos, like this one that's not playing for whatever, which basically just has you can literally see the exosomes as they're passing under the camera, and then the computer counts them, and measures the diameter, so we have a good estimation of the size and the quantity of the exosomes that we're passing under them. And we're seeing a nice big peak right at about 113, which is where we'd expect these type of exosomes to be, so we know that we're getting the right kind of particles in our isolations. Then we look for exosome marker presence to make sure that these are actually exosomes, even though they're the right size. We're looking for, these are two examples of two pretty common proteins that are on the membranes of these exosomes, CD9 and CD81. We're seeing them all show up in our samples. So we know that they have the exosome markers. And then we look at their morphology just to make sure that this process of collecting them hasn't damaged them or made them irregular in some way. This is a couple examples that we just got. We're seeing, we have control exosomes that are about the same shape. They're all rounded and they have about the same size that we would expect. And we're having the same in our patient exosomes. They are kind of varying in size, but that's pretty normal for exosomes. They're affected by a lot of things like osmolarity of the urine and a lot of other things in the kidney. So they can have a lot of varying sizes. Then, once we're done isolating and characterizing them, we're going to move on to our second phase, where we're actually doing the treatment. This is what we're right about to start in our lab. And for this, we're doing a proliferation assay or an XTT assay. Basically, we're going to have two different 96-well plates. One, we're going to seed with wild-type cell cultures, and the other, we're going to seed with PKD1 mutant cell cultures that serve as a model for the patient cells. We're going to seed them with all of the exact same population sizes in each of these wells, and then we're going to treat them with either no exosomes, exosomes from patient urine, or exosomes from control urine. We're going to let them grow for 24 to 72 hours, and then we're going to count and see how many cells are in each of the wells after these time points. So at each time point, we add the XTT reagent, and that basically only incorporates into live cells. So we can measure the absorbance, and then we know in each specific well how many cells are there now after the different growth time points. And so if we collect multiple different time points, we can get a graph that we're expecting to look kind of like this, where we're going to have different lines for our different treatments and for our different cell types that we're treating over time to see their proliferation and hopefully get different proliferation rates. And if we do, which we're expecting, if we're seeing that cells treated with exosomes from the disease patients are the ones that are growing faster, that supports our hypothesis, because the ones that are growing faster have the more cystic behavior. They're proliferating faster like cysts would. So for next steps, one, we just want to do a little bit more quality control on our exosomes. One thing that we haven't shown yet is that we want to make sure that, like the literature says, that these exosomes actually do have the two proteins that are implicated in PKD, PKD1 and PKD2. Those are a little bit hard to get to show up on a Western blot just because they're really big and they're in pretty low concentrations in the cells. And so instead, we're going to do proteomics analysis to make sure that we actually have those in our samples. And then while we're doing that, we can also make sure that these exosomes from different patients have comparable contents in them. So we're not getting a lot of variable contents inside of the exosomes. We're not expecting that they should, they probably won't, but we do want to make sure that that's the case. And then we can go ahead and actually perform these phase 2 cell proliferation assays. We're going to grow up control and PKD1 mutant primary cells. We're going to treat them with exosomes and see what their proliferation rates are. These are my acknowledgements. I can thank my lab, Dr. Chapman, Dr. Chen, and advice from Dr. West along the way, and funding for the Dr. Cook Foundation. Thank you.